Today, I will be discussing apoptosis in HL60 cells and how astragalus can affect this process. HL60 cells are a type of human leukemia cell that is commonly used for laboratory research because it continually increases in number, doubling roughly every 36 to 48 hours. This rapid reproduction made HL60 cells ideal for this experiment to see if the astragalus extract would affect this reproduction. In this experiment, we hope to see apoptosis rather than necrosis. Apoptosis would kill the cancer cell without harming other cells. Necrosis kills the cell but also releases that cell's contents upon the surrounding cells. And when it is a cancer cell that has died, it is not good to spread that cell's contents to the other cells. Astragalus is a Chinese herb that has been shown through research to have a substantial anti-cancer effect in human lung cancer cells as well as possibly other cancers. Astragalus can inhibit nuclear factor kappa B, which plays an important role in genesis, growth, development, and metastasis of cancer. Because of the anti-cancer properties of astragalus, I was able to hypothesize that astragalus can increase oxidative stress and induce apoptosis in HL60 cells. A cell viability assay was run for 24 hours using a control sample, a 70% ethanol sample, and an astragalus extract sample. Each sample also contained WST8. These samples were then run at 450 nanometers of absorbance in the spectrophotometer. The absorbances for 70% ethanol and astragalus extract were tested against the absorbance of the control. The astragalus t-test yielded a p-value of roughly 0.05, which means that it is significant. The significance of the astragalus sample suggests that it was able to induce apoptosis. A 24-hour morphology assay was run with the astragalus extract and HL60 cells. Cyto16 was used to observe the results under the confocal fluorescent microscope. Cyto16 highlights nuclear condensation as well as blebbing. Nuclear condensation takes place during apoptosis. The chromatin compacts and will show up brighter than the normal cells. There are a few cells in this morphology assay that appear to be undergoing nuclear condensation, such as these, and possibly others. Blebbing is when a protrusion or bulge sticks out from the normal shape of the cell. In this assay, it did not appear that there was any blebbing. Due to the nuclear condensation present, it is reasonable to conclude that this assay is showing that apoptosis is present. A COMET assay was run looking at the astragalus extract interacting with the HL60 cells. A healthy cell will still appear as a circle such as these. However, if the cell is unhealthy and the DNA has been damaged, the cell will appear as a comet because the damaged DNA will be pulled towards the anode. This is due to the DNA's negative charge. This cell appears to have the shape of a comet, meaning that this cell's DNA was damaged. The significant difference in my cell viability assay, along with the nuclear condensation in the morphology assay, and the comet visible in my COMET assay, led me to conclude that my hypothesis that astragalus can increase oxidative stress and induce apoptosis in the HL60 cells is supported. In the future, I think that it would be wise to test astragalus on other specialized forms of cancer, not just the HL60 cells and the lung cancer cells that they tested on in the other study. It would also be interesting to see if a more concentrated form of astragalus could produce better results. The sample I used was for human consumption as a supplement, so it is possible that the sample is not as potent as it possibly could be due to safety concerns. These were the references I used, and thank you for listening about astragalus and its relation to apoptosis in HL60 cells.